great deal of cell communication, and at this point you're wondering, as you have been ever since that day, how much of this, what's with the color? How much of this do we need to know? Does it look blue to you? Because it looks blue to me. Okay, so I'm not just freaking out. That's cool. I don't know what's happening there. The answer to that is I will make that very clear on the study guide that I send out to you today of how much of what Dr. Francis went through with you that you need to be responsible, or that you will be held responsible for. And it'll be very clear on the study guide that, again, will appear on Canvas sometime before the sun rises tomorrow morning. Now, what we're going to talk about together in cell communication, you can just anticipate, I mean, this is all coming. But again, the study guide will ask you specific questions from this. But there are only 18 slides. And again, you can anticipate pretty much all of this being on the study guide. I like this chapter. I like this chapter a lot. I like it a lot. Okay, so the first question we need to deal with is this one. What are 3 ways in which cells communicate? What are 3 ways in which cells communicate? And they basically, the 3 ways fall into one of 2 categories. Local and long distance. Local and long distance. And you all are probably too young to remember where there used to be a distinction when you would make a phone call between local and long distance, and you had to pay a ridiculous amount for long distance calls. And that probably inspired the authors to basically root these down into 2 categories. So the first type of local signaling. This is the most common. What's that? Did it? Oh, but now it's back to blue? Oh, there it's back to blue. So this is the first local signal that we're going to talk about because it is the most common local signal that cells will do. And this is where you have a controlling cell that will dump some product into the fluid surrounding that cell. Okay, and this product is called a local regulator because it changes cells in that local environment. Okay, and so this whole chapter, it calls it cell communication, but it's really just about chemical communication. Because cells communicate in really 3 ways, but we're only talking about one of those ways in this chapter, and that's chemical communication. So here, paracrine signaling. A controlling cell empties its product into the surrounding fluid, and that fluid changes neighboring cells. Probably the most interesting example of this would be cells that trigger an immune response. Okay? Where the cells will empty some kind of a protein signal that signals surrounding cells to induce inflammation. Okay? And then encourages other cells to send out signals to recruit white blood cells. Very good example of paracrine regulation. Now, the other form of local communication, local signaling, this is less common than paracrine. It's when a controlling cell changes its own behavior by releasing some regulator. And you're like, why on earth would it function this way, right? This seems so strange. It's like, why can't this cell just do what it's telling itself to do? Why does it have to tell itself in order for it to get done? And then you're like, wait a minute, I have to do the same thing, right? If I don't make a note, if I don't put a little sticky note on my mirror saying smile more today, tell someone you love them, I'm not going to do it, right? And so that's not why the cells do it, but I thought that was something that, because I read this and I was like, why would cells do this? And I'm like, I do this. Like, I write myself notes all day long just to make sure I get done what I know needs to get done, right? Be kind to your children today, you know? They're just kids, you know? Oh, man. Okay, so what's happening here is in an autocrine regulation, this cell is likely sending out a signal for cells in the surrounding environment as well. It just happens to also respond like those other cells do. So it's oftentimes this is done in addition to paracrine signaling. So at the same time that it signals everything in the surrounding environment to respond in a particular way, it responds in that way. Also, what we'll see in a little bit, there's a very specific pathway that cells go through to respond to a chemical signal. And so oftentimes this is the only way the cell can tell itself to do something, is to work with the way that cell is programmed. Okay? And it's programmed to basically bind, transduce, respond. But we'll see that in a moment. Bind, transduce, respond. Oh, that's good. Okay. And then so the other category, we've got local signaling, which are paracrine, autocrine, and then we have long distance signaling. And this is what we call endocrine signaling. Endocrine. Y'all have heard about the endocrine system, right? One of our organ systems. Endocrine system involves kidneys and several other glands, thymus gland, thyroid, okay? And so the endocrine signaling, a cell, a controlling cell, again, it's still releasing some chemical product, but this chemical product is getting into the bloodstream and is basically going to target tissues, but it's kind of going everywhere. And so this you see when like you have an adrenaline spike where your body releases norepinephrine and it gets into your bloodstream, and there are a lot of responses to it. Your heart rate increases, your airways dilate so you can bring in more air, your vessels all over your body dilate so blood flows more rapidly, and you're ready to go, right? And it requires a lot of different components getting involved. Great example of endocrine signaling. Endocrine signaling. Now, there's another one which y'all saw on your quiz, if you did the quiz prior, exocrine, which was not one of these three, but exocrine, the name suggests exactly what it is. Exo means outside, and so this is where you signal to someone else chemically what's going on. Humans, it doesn't seem that we rely too much on chemical cues, although there are some data to suggest that we do rely a little bit on chemical cues, and you might have experienced this when you walk into a room and it just feels tense. And it's like there's some data that suggests it's because you're picking up on chemical cues. Okay? But anyways, we rely a lot more on visual and auditory cues. But other animals rely a lot on chemical cues. And this is kind of fun. We had some good conversations. I had some good conversations with my children about this when we're taking our dog on a walk and he stops to smell a tree for minutes on end, and they're like, what is he doing? And I'm like, he's communicating. He's communicating with another dog that left a signal there, and he's responding. And they're like, ew. They're talking through pee? And I said, well, he's not talking unless he pees back, but yes, sort of. Yeah, Levi. Oh, yeah. Oh, yeah. You bet. Yeah, especially canids. Members of family Canidae, the dogs, very, very common. Also in cats and Felidae. But yeah, and then really common in insects to communicate using pheromones. All right. So to sum all this up, cells can also communicate through direct contact. That's not a chemical communication. Although, gap junctions can combine cytoplasms, and so you can move materials around. Also, when cells are touching each other, it usually sends, we've seen this before in the cell cycle, it sends a signal basically don't grow, right? You call that contact inhibition, and that's a form of mechanical communication. It's like when you have a puppy and you don't want it to grow, so you just hold it real tight all the time. But it doesn't work, it still grows, That was a terrible analogy. Oh man, I don't know why I said that, but it came to me, I had to say it. All right, and then cells communicate through local signaling, paracrine, autocrine. And so again, autocrine, the same cell sending the signal is responding to that signal. And for some of these cells, it's the only way they can get that response, is to actually produce what chemical they need to induce that response. And then cells can communicate through long distance signaling, hormones, and other materials are released. and carried in animals. You call this endocrine signaling. And then, so there are a lot of pieces of evidence to support that this plays an incredibly large role in the development of organisms. And so Earl Sutherland demonstrated basically the role between epinephrine, which triggers an adrenaline spike, and the glycogen metabolism, which again is very important to access energy stores. So oftentimes what you find here is with these endocrine signals, when you're doing long distance signaling, you're pumping something into the bloodstream and it can go everywhere, right? You put something into the bloodstream and it can go everywhere, which is part of the reason why having an infection spread to your bloodstream is so incredibly dangerous. Because once something gets into your bloodstream, it can go anywhere. And so with this, what you find is with the endocrine signaling, there's oftentimes a second necessary chemical component as well. And so it tends to help to focus it a little bit. So everywhere in the body might be getting that first signal, but there might be other places in the body that only certain places are getting that secondary signal. All right. So talk a little bit about the different forms of communication. We need to talk about, okay, let's say a cell actually does have another cell telling it something. How does it actually know what's being said and know what to do? And so to answer that question, we have to answer this one. What are the 3 steps in processing chemical signals? What are the 3 steps in processing chemical signals? Simply put, the 3 steps are reception, transduction, and response. Reception, transduction, and response. And so sometimes you'll see binding, transduction, response. The nice thing about saying reception is it has the same root as what protein component? Receptor. Okay, so it tells you these roles of these protein receptors. Way long ago, maybe the 2nd or 3rd week of our semester, which is like 3 months ago, we talked about proteins in the cell membrane, right? And one of those proteins are receptors. And a lot of these receptors function in chemical communication. And these receptors, they are perfectly designed to bind a very specific material, a very specific signal molecule. And so the beauty of this is it allows your body to create cells that have that receptor that will respond to that signal, and at the same time to create cells that do not have that receptor, therefore cannot respond to that signal. Okay? And it's a beautiful thing, because you're already thinking, like, we've got this endocrine signaling, those signals are going everywhere. How do we ensure that everything isn't going to respond? Well, one of the ways you can do it, you only have the cells that need that receptor actually transcribe and then translate that receptor. Okay? And then transduction. Transduction it's basically a translation event. It takes this signal from this signal molecule bound to this receptor and translates it into an intracellular chemical signal. And what's interesting is just like when you're translating from one human language to another, when you're translating from one chemical to another, oftentimes you can get the same pathway in here from different signal molecules. There might be multiple ways in which you can get this same process. Okay? That was a little bit of what Dr. Francis was talking to you about with his G protein coupled cell receptors. That's good stuff. All right. So in order for it to respond, it has to bind the signal. We call this reception. Now, if the signal molecule is polar, where's your receptor? If the signal molecule is polar, like a protein, where's your receptor? Outside. It's on the cell surface. So receptors for polar molecules have to be on the cell surface. But if your signal is nonpolar, It's one of your hormones, one of your lipid hormones. So it's nonpolar. Where do you think you're going to find those receptors? Oftentimes inside the cell, because those are materials that can diffuse across the plasma membrane, right? If they're nonpolar, they can diffuse across the plasma membrane. So first you've got to bind the signal. Then you've got to translate the signal. And this is transduction. And the reason why we give it a special name, you're like, why can't we just call it translate? Well, it's like translation has a very specific meaning in cell biology, right? What is the meaning of translation in cell biology? Changing what? You take RNA and you translate it into an amino acid sequence, into a polypeptide. Okay, so translation has a very specific meaning in biology. And then on top of that, translation has a very specific meaning in our common lingo. When you think translate, you're like, give me a sentence in this language and I'm going to translate it directly into this next language. It is way more complicated than that. It is way more complicated than that, because what happens during this process of transduction is we have a whole series of reactions that take place to get some chemical that we need for the response. Okay? There are several steps in this process. And you're like, why is that the case? It should be much simpler, right? It should be much simpler. We bind a chemical signal, we immediately translate into something internal to the cell, and we get a response. Hold on to that, because we're going to talk a little bit about that during our lecture break, which will come shortly. And then the last thing the cell needs to do is it needs to act. It needs to execute some response. And so specific molecules within this cell that appear after that transduction pathway, that signal cascade, they get some action from the target cell. They get some action from the target cell. Here's a statement that came right out of your textbook. I thought it's an interesting one that we'll also talk about during our lecture break. The idea here is that unicellular organisms, they communicate. So bacteria, individual bacteria, communicate with other bacterial cells. They do so when they make biofilms and others. A number of other single-celled organisms communicate to assess relative population size and even to create some simple division of labor. And so the idea here is that cellular communication evolved prior to the evolution of multicellularity. And some additional support for that is that a lot of the protein components that you find in eukaryotes, like those receptors for binding specific signal molecules, you also find in prokaryotic cells. Like the same proteins involved in this communication, this chemical communication. All right? So here's what we're going to do. We're going to take a lecture break, and I want you to do two things. One is I want you to come up with at least one reason why this transduction would be more than just a simple one-step process, but instead would require several individual reactions. One reason why that might be the case, okay? And we'll talk about that. Does that make sense? Okay, and then the other thing I want you to do is come up with another reason why single-celled organisms would need to communicate, other than that their ability to communicate basically facilitated giving rise to multicellularity. Okay, because the biblical story of origins is inconsistent with the idea that multicellular forms evolve from single-celled forms. Okay, that's not the creation account that's represented in Scripture. Yeah, Tara. Yeah, the first question is to explain one benefit or one explanation why transduction requires a series of reactions rather than just a single reaction. Okay, and then the other one is to explain why do unicellular organisms need to communicate? Why does it make sense that they would communicate and not then be used as evidence that multicellular forms evolved from single-celled forms that were capable of communication? Why do single-celled forms need to communicate? Is 2 minutes enough time? Okay, 2 minutes, we'll begin this discussion, we'll continue it. 2 minutes, starting now.
I think because evolution is true. What's up? That's the
That means we talked about getting certain molecules that trigger certain reactions, right? Like when you're doing translation of bioenergy or whatever, we talked about certain molecules that activate other molecules by changing reactions, right? So I'm kind of thinking that during translation that happens also. It's like the shapes of the other cell, like this molecule comes in and changes the shape of the cell.
It changes like the protein or whatever.
Yeah, and then that activates the protein, thereby triggering a certain behavior in that cell. So I just kind of feel like this molecule comes in and it has a specific law.
That's used like ATP too, right?
Yeah. Yeah. Wow. Wow.
Yes, Dan.
All right. Do we have an explanation for why this process of transduction involves more than a single reaction? Yeah, Levi.
Because with each step, we can increase the magnitude.
Yes, yeah, absolutely. And that's a big reason why. Is every time, let's say you have a single reactant that can trigger the response of several products. And this, oftentimes what you see is that this first step in the transduction pathway is to activate an enzyme. And we know enzymes are not used up in chemical reactions, right? So an enzyme can facilitate that reaction 100 times, even 1,000 times. And so when you turn on an enzyme, it increases, it multiplies, it magnifies whatever that signal is to ensure that the cell responds and responds properly and efficiently. Good. Yep, that is one big reason why. Micah.
Could there be a check that takes place to determine whether or not you need this?
Absolutely. Yep, it gives you time to determine whether that response is justified, right? So if the cell gets it binds a signal molecule that says basically eventually execute this response, but then that signal molecule disappears, there's not very much of it, gives the cell a layer of control and an ability to abort a response, okay? Which you don't have if that transduction is a single reaction, right? You're like, I don't know, I've taken Gen Chem lab and I know even single reactions can take a long time, right? But that's without a catalyst. Single reactions with a catalyst like an enzyme, they don't take very long. They don't take very long at all. Yeah.
About amplifying the signal. Since it carries the signal, so you need it to.
Absolutely. Yep. And that's yeah. And that's absolutely a part of it as well. Amplify that signal for sure. Okay, good. What about the second question? Why do unicellular organisms need to communicate? This one's more interesting. Yeah, Tara.
That they need to be able to recognize each other. That's not a forward.
Yeah, what's interesting is all bacteria seem to be able to determine not just that there are other bacteria present, but whether or not they're closely related to them. Whether or not they're the same species, and even if they are the same species, whether or not they're closely related, which is really interesting. And a number of bacteria make colonies and make complex biofilms, and some of those are necessary for us to survive, right? Talked a little bit about these biofilms earlier. No, last semester. Last semester we talked about biofilms. And a lot of these biofilms are really necessary for us to ensure that we've got a a stable and safe barrier from infection. A lot of that is actually done by what we call mutualistic bacteria, or our normal flora, creating a biofilm to protect us. And then what's kind of interesting is we're starting to find a lot of evidence to suggest that we control our normal flora in more ways than just like accidentally being exposed to it. That our tissues actually communicate with our gut bacteria, okay, and hold them in check, okay, which is really interesting, gentlemen, okay, which is really interesting. And so there's a lot of evidence, and if we're going to communicate with prokaryotic cells, we better communicate with them in a similar way to how we communicate with our own cells, right? And again, there are a lot of data to suggest that we actually do this, and more than we ever imagined, that our cells are communicating with our gut bacteria. All right, next question. How does this cell know which signal molecules to receive? Because again, it seems to be that we just have, with this chemical communication, something that is very unfocused and almost random. And so here, we can talk about, I mentioned this already, but these receptors, they are proteins. And we know that proteins are made up of amino acids, yes? And we know that amino acids have very specific chemistries because we talked about different types of mutations, right? We talked about silent mutations. Somebody remind me what a silent mutation is. Yeah, Levi. Yeah, why? Yeah, and it's exactly the same. So it's a mutation in the codon that still gets translated into the same amino acid. And then we have missense mutations, where the missense mutation, what happens there? Yeah, it changes the amino acid, right? So that you've got a mutation in your codon that gets translated into a different amino acid. Now, some of those missense mutations translated into a chemically similar amino acid, so that the protein still behaves the same. So we know that proteins have very specific sequences and behave in very specific ways because of their chemistry. And so what you should anticipate is, just like enzymes are very substrate specific, right? Do you remember talking about that with enzymes? They are substrate specific. They are chemically and physically designed for a very specific substrate. The receptors are the same way. They are chemically and physically designed for a very specific signal molecule. They are very specialized. And so how does the cell know which to respond? It's the controlling cells release polar signal molecules, and this is just talking about the receptor changes. Okay. This is the answer to our question. Different cell types produce different combinations of receptors. So different cell types produce different combinations of receptors, so that the various cells only produce the receptors for the signal molecules they need for their own function. And what you find is, even in a specific cell line, the receptors that cell produces varies through the life of that cell. So early in the cell's life, when it's still going through this development phase, it responds to signals differently than what it will do later in life, and to different signals. Okay. And then, yeah, Davies.
So, for example, for pain receptors, for a kid that has not developed fully, would the response be different as compared to an adult?
Yes. Mm-hmm. Yeah, you're talking pain receptors?
Yeah.
So pain receptors are a little bit different also because pain receptors are sensory structures. They're modified neurons to develop a sensory structure. So they're a little bit different, but they tend to still behave differently. But this, we're talking more about like chemical receptors, specific membrane proteins. And so how does a cell respond to binding a signal molecule? Well, binding is step one, right? Reception is step one. Step two is transduction. And so here is a wonderful example of a transduction pathway. Not as good as the example Dr. Francis gave you, but still a really good example of a transduction pathway. And so in this one, it's a little bit more general. It basically, as the receptor binds that signal molecule, what this receptor does is activate this enzyme. You see it activated the enzyme, and how did it activate it? It phosphorylated it, right? And what do we call enzymes that add phosphate groups to things? Kinases. And so this type of receptor is oftentimes what we call a kinase receptor, or a receptor kinase. Okay? So it's a receptor and it's also a kinase. It phosphorylated this enzyme and activated it. And then when this enzyme is activated, it activated this enzyme. And how did it activate this one? It added a phosphate group. And what do we call enzymes that add phosphate groups to things? Kinases. So this is also a kinase. And then this one phosphorylates. This is a kinase. And this one phosphorylates. That's a kinase. And we have this whole series of kinase enzymes that activate other enzymes down the way by phosphorylating them. And so a lot of times what you see is this transduction cascade, or this transduction pathway, is referred to as a kinase cascade. Okay? So that's a term you should be familiar with, and that's a term when you see kinase cascade, you need to think we're talking about a transduction pathway. A series of enzymes, a series of kinase enzymes that are phosphorylating the next enzyme in the pathway. Yeah, Micah?
Is that amplification?
Yes. It's amplification, but it's also what you're talking about, giving you a level of control. Because if you have to phosphorylate this enzyme to activate it, how do you deactivate it? If when you add a phosphate group to this, it activates it, now you have an active form of this enzyme, how do you deactivate it? Remove the phosphate group. And what kind of enzymes remove phosphate groups? Phosphatases. So now the cell can actually deactivate this enzyme before it finishes this whole kinase cascade. Because oftentimes it has to do this entire process to make this that gets some cellular response. And so any place along the way, a phosphatase can deactivate this enzyme. And so it gives the cell a level of control to abort the response before it even happens. But it also gives us the ability to amplify it, what Levi and Davies were talking about. Yeah.
How long will a reaction like this usually take?
It depends on the complexity of the response. If the response is incredibly complex and it requires the cell to invest a lot of energy, it tends to take longer to do this, to give the cell more control over whether or not it should actually happen.
So, but is it
You're usually talking about in a matter of minutes,
okay?
and not like days or, you know.
For seconds.
No, and it's not usually seconds, it's usually minutes. Because you usually have to build up a high enough amount of these enzymes before you actually start to get a significant transformation of the next in the process. Yeah, that's a good question. And then here's exactly an illustration of that amplification. Okay, so here we started with our receptor. The receptor activated 1,000 of these enzymes. Each of these enzymes activate 100 of these. So now you have 100,000 of these. And each of these activate, what would that be, 100 of these? 100 of these. And so now you have 10 million of these. And this is what you need to actually get the cell to do some response. So it started with a single signal molecule, and now you have 10 million of your active components that are necessary to get the cell to carry out some task. Okay, and so here I want to show you some experimental research. So here we've got this RTK. Do you see that? I know it might be small, and it is small. I don't know how well you can see it. This stands for receptor tyrosine kinase. It's called a receptor tyrosine kinase because it's a receptor. It's got these tyrosine residues, and it's a kinase. And if it's a kinase, what's it going to do? It's going to phosphorylate an enzyme and activate some enzyme. And so here you have this receptor in its resting form, okay? And then when these receptors bind your signal molecules, it induces them to dimerize for those two receptors to link together. And when they do, it activates these protein kinase regions, and then those will get their phosphate groups, and then they will phosphorylate other enzymes, okay, and activating those enzymes. And so it gives you a little bit of the molecular aspect of what's actually happening here, okay? So this receptor in its resting form, it's not doing anything. But once it binds that signal molecule, it dimerizes, it activates the phosphorylating regions, the kinase regions, gets some phosphate groups. And where do you think these phosphate groups come from? ATP. Okay, so it's requiring some energy, and now these kinase regions can phosphorylate an enzyme. Now notice the different regions on here can phosphorylate different enzymes. And so now you have the same receptor with the same signal molecule that can induce two different transduction pathways, two different cellular responses. Because maybe the cell needs to respond in two ways to that. One, maybe grow and divide. And then the other thing, create some signal that's going to tell surrounding cells to also grow and divide. Make sense? Okay. All right. So to sum all this up, how does the cell respond to binding a signal molecule? It transduces that signal. So I've got a couple of questions for you. Or I've got this question. How can a molecule that doesn't enter the cell cause so much change in that cell? Okay? This signal molecule is never entering, but it's going to completely transform that cell. And it's all because of the amplification of that signal. Okay? These kinases and this kinase cascade creating a chain reaction. Okay, and then ultimately you are going to, at the end of this, trigger some target protein, and every step along the way can amplify this response, and every step along the way can be controlled. Here's a second question. How does this process stop? How does the cell know when to stop this process? Tara? Yeah, a lot of it's negative feedback. That end product, it's being made, a lot of it's being made to execute some response, but it also acts as a feedback on some earlier part in the process. Just like ATP acts as a inhibitor of reaction 3 of glycolysis. So does pyruvate. So if you've got a lot of ATP and a lot of pyruvate, it inhibits an earlier step in the process. Okay? And then the cell can also know when to stop when it gets a second signal telling it to stop. And then a phosphatase can go and pull the phosphate group off those activated enzymes. Yeah, many of these receptors are receptor tyrosine kinases, and they have built-in kinases. They are both receptors and enzymes. And then, like we've already mentioned, phosphatases function as off switches. Endocytosis can also function as an off switch. How do you get a protein receptor out of the plasma membrane? Endocytosis. You bud it off into a vesicle inside of the membrane, and then you can go and degrade it. Okay, and this can act as a permanent off switch. Okay, when you eliminate that protein receptor. Phosphatases act as like basically an immediate and temporary off switch, because then you can phosphorylate it by a kinase and reactivate it. But you pull those receptors off the plasma membrane, and that acts as a permanent off switch. The only way to counteract that is to make that protein receptor again and put it back on the cell membrane. Yeah, David.
So the cell in endocytosis will remove the protein?
Yep, and oftentimes that's part of the maturation process of the cell. So like the receptors that it needs early in development, it doesn't need later in development. And so what it'll do is once the cell gets to a certain point in its process, it will remove those receptors from the surface. And so then it is no longer sensitive to whatever that signal is. Now, what's interesting is you'll have cancer cells that one of the things that they'll do, oftentimes in many cancers, is they'll remake those receptors that are present in early of the cell that basically get it to go through the cell cycle, so they become very, very sensitive to signals to tell them to grow. All right. Any questions? So this is the last slide. And so I have nothing more to give to you for today. I will put, in addition to getting your study guide up on Canvas before the sun rises tomorrow, I will get a quiz for Monday up on Canvas, either tonight or sometime tomorrow, and it will start going over regulation of gene expression that we're going to talk about next week in class.